Right. Hello, everyone. My name is Spyros. I'm a PhD student at the MRC University of Glasgow Centre for Virus Research. And today we'll be talking to you about our research on exploring the origins of SARS-CoV-2 in the light of recombination. So I'll start off by explaining why it's crucial to account for recombination when exploring the evolutionary histories of viruses in general, but particularly this group of coronaviruses. So imagine a yellow virus 1 and a blue virus 2. They find themselves in the same cell at the same time and they recombine. So now, if you were to take the entire whole genome of these two viruses and infer a phylogeny, that phylogeny would not really reflect the true evolutionary history of the viruses. What one needs to do is determine where the recombination breakpoints are in the genomes and then split the alignment by them to create non-recombinant, putatively non-recombinant regions, and then infer phylogenies out of them. So then you'd see that, for example, blue virus 2 is more closely related to green virus 3 for this region and kind of vice versa for the other one. So what we did is retrieve a set of 78 sarbecoviruses. This is the subgenus that SARS-CoV-1 and SARS-CoV-2 are a part of. And these are essentially all the sarbecoviruses we have a whole genome for. And we did this preliminary recombination analysis with RDB5 to essentially find all the kind of unique short recombinants in each sequence and take them out of the whole genome alignment and to kind of determine a preliminary phylogeny that's essentially not recombinant. And you can see that SARS-CoV-2 sits here within this nCoV clade, along with about 20 other viruses. And this clade is phylogenetically distinct to this non-nCoV clade that has better sampling. It has about 50 or 60 viruses, and SARS-CoV-1 is a part of this as well. So then we moved on to using this software GUARD that essentially determines recombination breakpoint positions for the entire alignment. And then you can essentially split up the genome into putatively non-recombinant regions by them, similarly to how the cartoon explained before. So we determined 22 recombination breakpoints with GUARD, and we were able to split up the alignment into 23 non-recombinant regions and then infer the phylogeny for each one of these regions. So I'm going to go through what the closest known relatives to SARS-CoV-2 are. So RATG13 is kind of the first one that came out, followed by RMYN02. And then very recently, these two viruses, RPYN06 and PRC31, have been published. And these genome schematics here explain exactly what I've been talking about. So the genome is split up into 23 putatively non-recombinant regions. And then the colors correspond to how closely related each virus is to SARS-CoV-2 for each one of those non-recombinant spans. And then the deeper pink, it means the closer to SARS-CoV-2. And for blue regions, it means that this virus for this region is actually clustering in the non-nCoV clade closer to SARS-CoV-1. Now, if we look at one of the kind of longest non-recombinant regions, for example, region 5, and zoom into the phylogeny, you can see that RATG13 sits here, SARS-CoV-2 here. And then these three viruses form this Yunnan clade that is essentially the closest clade sister to SARS-CoV-2. And I should mention that all these four viruses have been sampled in Yunnan, China, but there's other closely related viruses that have been sampled outside China, namely RAX203 has been sampled in Thailand, and these RSH-STT viruses sampled in Cambodia. And you can see that there's also evidence of between clade recombination in these viruses. So if we look at region 4, for example, you can see that RAX203 seems to cluster within the Yunnan clade that I just showed. Whereas for region 10, the RSH-STT viruses pair up with RATG13. And here you could argue that SARS-CoV-2 is actually recombining out of this clade. But what I'm really trying to show here is that each one of those non-recombinant spans across the genome infers a different incongruent topology for these very closely related viruses, suggestive of recent recombination between them. And we can basically tell how recent that would be. We can put some dates on it. This is our beast dating analysis for region 5. And you can see that we estimate the Yunnan clade to have shared a common ancestor to SARS-CoV-2 about 40 years ago, followed by RAG13. And then these two viruses sharing a common ancestor to SARS-CoV-2 less than 100 years ago have been sampled in Zhejiang. This is a province of China on kind of the other end of the country. And we can look at a map as well. So SARS-CoV-2 emerged in the city of Wuhan in Hubei, central China. Then Yunnan sits here with these 4 viruses I mentioned. And then Zhejiang is right across to the east side of China with these viruses, and then Thailand here and Cambodia there. And this is a significantly extensive geographic range that these closely related viruses that have shared ancestors in the past century or 2 centuries have been sampled across. Now you could argue that they're dispersed, but maybe there's clustering in the hosts that these viruses have been sampled in. And apart from 2 virus lineages that have been sampled in pangolins and obviously SARS-CoV-2, all the other viruses have been sampled in horseshoe bats of the genus Rhinolophus. However, you'll notice that basically almost every single one of these has been sampled in a different species of the Rhinolophus genus. We can look at the geographic ranges of these species. For example, Rhinolophus pusillus and Rhinolophus affinis have very wide geographic ranges that essentially extend across the wide geographic range that these viruses have been sampled in. And some other horseshoe bat species that have had these viruses have slightly shorter geographic ranges, but you can see overlaps between them. So for example, Malayanus is mainly found in the Chinese peninsula, but it's also found in Yunnan in China. So essentially, it looks as if these bat host ranges are overlapping between one another and also overlapping with the sampling locations of these closely related viruses. And this is suggestive of these viruses being able to jump between these bats quite easily and then extend across and move across the geographic ranges of the bats. So I'm going to just quickly finish off by showing you this illustration from our recent perspective that clearly shows where these viruses have been sampled and how extensive this geographic range is. So to summarize, in the last century, it seems like there has been ancestral recombination and wide geographic movement of these viruses across China, the south of China, and kind of Southeast Asia. And this wide geographic region perfectly overlaps with the host ranges of the bats that these viruses are found in. Now, we still haven't sampled the population of the proximal ancestor of SARS-CoV-2, and the closest kind of virus clade that we know of is the viruses sampled here, PRC31, RPY06, RMY02, that shared a common ancestor to SARS-CoV-2 about 40 years ago. So this is still quite a long time, considering that the movement of these recombinant viruses in the past century. There's clearly big gaps in the phylogeny, and it seems that the only way forward to getting closer to knowing the origins of SARS-CoV-2 is really increasing wildlife sampling across this entire geographic region. And this is definitely not a trivial effort. So with that, I'd like to thank everyone involved in this work. Joseph Hughes, David Robertson, the labs I'm part of at the CBR, Darren Martin and his team, Sergei Pond, and Wei Xia and Xiaowei Chang. And I'd like to thank you for your attention. I'm happy to take any questions.